Hi everybody, this is Dr. Polito and this is my majors biology lecture on cellular respiration. This PowerPoint should match up with what you have online. If it doesn't, that means that you're watching this in the future. The first few slides I'm glossing over. These are just silly slides that we go over in class. Okay, this slide here just tells you what a calorie is. So the context here is we're talking about energy transformations in our cells, and this just provides some context. So I'm going to gloss over this too. This slide I'm going to gloss over. This is just telling us the differences between cell respiration versus when we generally say respiration, we're talking about breathing in and out, as opposed to extracting energy from organic molecules. And so we're going to talk here now about the general concept of cell respiration. All right, so when we say organic compounds, we're talking about things like sugars, we're talking about things like proteins, and we're talking about things like lipids. Okay, notice I'm leaving out nucleic acids. That's because most of the time nucleic acids, we don't generally try to get energy out of them because they're too toxic. They have too much nitrogen in them, basically, to keep it simple. So we're going to take these organic compounds, and in the presence of oxygen, we are going to pull the energy out. We're going to oxidize it, right? We're going to pull the energy out, and what's going to be left is some carbon dioxide and some water. Okay, so we breathe in oxygen, and at the end of the game that we're going to play here, oxygen is going to grab some low energy electrons, and together with a proton or two, we're going to create some water. Okay, so this right here, this energy, this is the energy that used to be in here. Okay, so we've just pulled it out basically. Now for glucose, which is what we're going to use as an example to all this, when you take the amount of free energy in glucose and you calculate it out, it comes out to exactly 686 kilocalories per mole. And that's an ideal. Okay, we're obviously not going to extract all that energy. Nothing's 100% efficient, of course. All right, so another way of saying this is that cell respiration is basically a series of redox reactions. Here's another slide showing the more specific example of glucose being broken down, right? So specifically, we would say glucose is oxidized to carbon dioxide, while oxygen is reduced to water, okay? And then another simple way of looking at this is we talked about metabolism being the sum of the catabolic and the anabolic reactions, right? So in this case, cell respiration is the process of catabolism where we're going to break down that high energy molecule into its low energy constituents. And in the process, we're going to take that energy and put it to work by putting the energy into these high energy ATP molecules, which we will then use to do our work. So as ATP falls back downhill, the energy gradient to ADP, we're going to use that fall to couple with the building reactions, the anabolism. Okay. Now this silly slide here just shows us three situations where energy is being released from a system pretty quickly, right? Think about, Mr. Bean there eating a pair of chicken wings really fast, Cookie Monster eating his cookie fast, or all the gasoline in a car being released all at the same time, exploding at once, right? So the idea here is that when we have energy stored in a system, it is much more efficient to take that energy out bits and pieces at a time, slowly, right? And this is something that's pretty intuitive in our everyday lives. If you want to do something right, you do it slowly, carefully, and you make no mistakes. All right, so that's the idea of why there's multiple steps. To understand how energy gets shuttled around the cell, in other words, how we're going to grab the energy from glucose, extract that energy and then put it into something like a hot coffee cup, and then move it physically to go do some work somewhere else with it. So let's make sure we understand a few of these basic concepts here. The first is understanding what NAD is. NAD, nicotinamide adenine dinucleotide, is, as that last letter implies, a dinucleotide consisting of two sugars, remember there's the ribose, two phosphate groups, and then two nitrogenous bases. In this case, we have nicotinamide, which the cell synthesizes from vitamin D3, I believe, and adenine. Okay, And so this dinucleotide's job is to stably hold very high energy electrons. It's a hot plate. That's the way to think of it. Okay, and it is given the energy by some enzyme that rips the energy from a substrate and hands it over. The enzymes that do this are called collectively dehydrogenases. As that name suggests, it gets rid of hydrogens from the substrate. So we're going to take 2 hydrogens from the substrate. So this could be, glucose, whatever. And we're going to steal 2 hydrogens from it in total. So really what we're doing though, okay, we're going to take 2 electrons and we're going to take 2 protons, right? Well, what do you mean by protons? Remember, if we take hydrogen, which naturally exists as H2, 2 protons surrounded by 2 electrons whizzing around, right? So this system here, this is 2 protons and 2 electrons, right? And so another way of saying a proton, you could think of that as just a lone by itself hydrogen atom, right? So H plus is interchangeable with P, with protons. So when we say H plus, like you see here, that's just a lonely proton, okay? So what dehydrogenases do is they take their substrate and they strip it of a pair of hydrogens and they transfer that stuff to a substrate. In the case of NAD plus rather, we're going to take a pair of electrons, okay? So 2 electrons, these are the high energy electrons that we've been playing with since day 1 on the photosynthesis lecture. They're just continuing their story here. These 2 high energy electrons are going to find themselves in NADH, or NAD, okay? And so if we take 2 electrons and a proton, we're going to get a hydrogen. So there's your NADH. Right? Now, because dehydrogenases rip a pair of protons off, or rather a pair of hydrogens from their substrate, there's going to be another hydrogen around, which literally just gets released into the solution. And that's why sometimes you see this. Okay? It's not really necessary. This is not necessary to the story. It's not important, but just understand it's there. That's because that's how these enzymes work. Okay? So, NAD plus gets reduced to NADH. Okay, so this is a reduction step. We would say that the initial part, the initial form of NAD plus is the oxidized state, and the form with the energy is the reduced state. And this is the product we're looking for. These are the important molecules that we're going to get and use to eventually make some ATP out of. Okay? So in addition to nicotinamide adenine dinucleotide, we're also going to run into its little cousin, FAD, or really FADH. Wait, no, you know what? Let's be right here. It's FAD. It just doesn't have a positive charge. And FAD is flavin adenine dinucleotide. All right? And so what FAD, like NAD plus, is going to do, it's just going to, by some other enzyme, it's going to receive all that electrons and protons. And in the case of FAD, it gets both of those. So it's FADH. Okay, in the case of the NAD, we just get NADH, right, and plus the extra proton. Okay, so these are the final products of this whole concept. These are the energy carriers. These shuttle the energy from glucose. Let's review the mitochondria very briefly. The mitochondria is an organelle inside all eukaryotic cells, and it has 2 membranes, 1, 2, the smooth outer membrane, and the very convoluted, folded up inner membrane. And each of these little folds here, these finger-like projections, we call cristae. Okay? Also recall the mitochondria has its own DNA in there. There's the mitochondrial DNA. It has its own ribosomes, right? But those are not important to the story right now. But that's the basics of the mitochondria. And remember that they divide by binary fission. Right? What's so special about these? What's special about these is that, again, in the presence of oxygen, we're going to take that pyruvate that is the product of glycolysis, and we are going to transport that pyruvate into the inner matrix here. That's the matrix is the inner space of the mitochondria. And we're going to transport the pyruvate there for further breakdown to get more energy out of it. So let's go through some details now. Here's an overview of the process, and what I'm going to do here is I'm going to spend just a minute giving you a very brief overview of what we're going to dig into. So the first thing I want you to do is step back, and I want you to put this stuff in 3D space. If this is a cell, okay, here's the nucleus, here's the mitochondria, okay, well, all the stuff we just talked about. And the first thing we're going to talk about, the first mechanism, is when sugar, glucose, gets transported into the cell, into the cytoplasm. It goes through this first process called glycolysis, which literally means sugar break, right? So we're going to break the sugar down. Then, and so we're going to talk about this. This is glycolysis in a nutshell. And then from there, in the presence of oxygen, so long as oxygen's present, we are going to find the breakdown products that's left over, the pyruvate. We're going to move that into the mitochondrial matrix, where it is going to be further processed. Okay, and so the first step that happens after it goes in there is it gets oxidized. And so the next step here is called pyruvate oxidation. Just a simple pathway here. And then the products of that get fed into the Krebs cycle, which also occurs in the mitochondrial matrix. Once the Krebs cycle is complete, the entirety of that original glucose molecule, all 6 of those carbons, are now given back to the universe. Here are the 4 that are produced from the original 6 of the Krebs cycle, of the original glucose. And then the other 2, those get released earlier in that pyruvate oxidation step. Okay? And then the last thing that's going to happen is we're going to collect all of our high energy carriers we just talked about, the ones that are produced in the Krebs cycle and the ones that are produced back in glycolysis. And we're going to take all of these and we're going to use that high energy pair of electrons in each of these and pump that energy into a transport chain that we're going to see resides in the mitochondrials, the inner membranes, in the inner membrane. And it's going to work in a way that's analogous to how we talked about photosynthesis. We're going to pump protons across a membrane, and then we are going to use that proton motive force as part of ATP synthase to make some ATP. Okay, so we borrow these concepts here from the photosynthesis that we talked about already. So that's the overview. Okay, glycolysis, pyruvate oxidation, the Krebs cycle, oxidative phosphorylation. That's the pathway that we're going to talk about. This is all in the presence of oxygen, okay, as we'll learn about. In the absence of oxygen, and we'll talk about this at the end here, you have a process called fermentation. Okay, so there we are. There's a basic roadmap. And this is a simpler version of it. What this is doing is only showing you the products of each of the reactions that we care about to make some ATP. All right, so this is a very plain picture here. And so all I want to say is at the end of glycolysis, we have some pyruvate. The pyruvate then goes and breaks down into an acetyl-CoA, which we'll learn what that is in a bit. Okay, and then that goes away in the Krebs cycle, and all that's left is the energy latent in the NADH and the FEDH2 that we want to track again, that we're then going to pump into oxidative phosphorylation. Okay, so this is a simplistic view of how we get the ATP out of the rest of the system. Okay, so we're going to extract all the energy from pyruvate, convert it to acetyl-CoA, burn the acetyl-CoA, and in all those processes, we're going to build up these high energy carriers where we get the big payoff here at the end with oxidative phosphorylation. All right, so let's talk about glycolysis. Glycolysis is the most ancient of the metabolic pathways we're going to talk about. And this is common to every single one of the domains of life we've talked about throughout the semester. The archaeans, the bacteria, the eukaryotes. Within the eukaryotes, the fungus, the animals, the plants, the protists. Everybody has within their little domains in the subtaxa cells capable of performing glycolysis. There are very, very few exceptions. Okay. Glycolysis is simply the hydrolysis of a 6-carbon glucose. Hydrolysis, remember, breaking it down. So this is a catabolic reaction, right? This is it's releasing energy. It is spontaneous, right? we're going to break down a 6-carbon glucose molecule to 2 3-carbon pyruvates. Okay? And in order for this to happen, we need a place to put that energy as we extract it. And so that's why we say it requires this empty bucket, the NAD+, right? The oxidized form of this molecule that can then receive the energy from the glucose. And glycolysis is divided into 3 major phases, consisting of a total of 10 chemical reactions. You are not going to need to know the details of the 10 chemical reactions, but you do need to know the 3 phases and the concepts here. Okay, so the first phase, phase 1, we call the energy investment phase. In this phase, we need to put a little energy in, so we're going to add 2 ATPs to the system to get the ball rolling, so to speak, right? You have to spend a little money to earn a little money. So we're going to add some ATPs here, okay? The second phase, we're going to, once we get that energy in there and create this unstable intermediate, we're going to break that 6-carbon molecule into 2 3-carbon molecules. And then from there, we're going to take those 2 3-carbon molecules and extract the energy from them. And that's that last phase, the energy liberation phase. Okay? So overall, we're going to put 2 ATPs into the system. Okay? We're going to need 2 NAD pluses to carry the excess energy we're going to talk about in a second. And what we're going to do is we're going to release the energy, and we're going to produce at the end of it 4 ATPs, and we're going to have 2 NADH molecules. We're going to be able to reduce that. All right, so we make a gross amount of 4 ATP, but because we spent 2, we say we net 2 ATP. Okay, you spend 2 ATP to make 4 ATP. Right? If I give you $2 and then you give me $4, I just made $2 profit, right? That's the concept. So here is the first phase. The first phase, the energy investment phase, is a series of 3 enzymatic reactions, mediated each separately by 3 different enzymes that we don't have to worry about. And in this process, these enzymes take ATP and they add those phosphate groups to the glucose molecule, creating a product called fructose 1,6-bisphosphate. And this fructose 1,6-bisphosphate is a very unstable molecule. So basically, we put energy into the system to get glucose positioned in a way that it will easily start rolling downhill. The next phase, the cleavage phase, these are reactions 4 and 5. This just takes the very unstable molecule and begins to break it apart. Okay, and so we have these 2 intermediates, these 2 very high energy molecules that happen to have the same name as something you should already be familiar with. This is glyceraldehyde 3-phosphate, right? Where'd you see that before? Think about it. If you said photosynthesis, you'd win. Right? So this is the product of photosynthesis. And so now here we are getting it back out. The last phase of glycolysis consists of a series of 5 chemical reactions, steps 6 through 10, in which we now take that G3P and what is essentially a reverse of the photosynthesis pathway, extract the energy back out. Okay, so if you remember, we had G3P, right? Then you might remember BPG, and then you might remember 3GP, right? So these 3 here, if you remember the Calvin cycle, this is the this is what happens after you take that ribulose bisphosphate and you add the carbon dioxide to it, and then you pump it full of the energy, and you get this guy, right? And so this is all just the opposite. And so, because remember, the second law of thermodynamics tells us you can't have any energy transformations that are 100% efficient. So of course, this isn't exactly a reverse, right? In the photosynthesis pathways, remember we had NADPH, right? This molecule here is much higher in energy than the littler cousin NADH, right? One way to think of it is plants have the phosphate, plants have the P here. So this molecule here holds more energy than this. So when we get the energy back out after photosynthesis, we're not getting it back in the full way we used to. We're getting it in a slightly weaker form, right? So we're still technically going downhill. Okay, so in these 5 steps, we're going to yield a total of 4 ATPs and 2 NADHs. Okay, why all this? Because remember, glucose broke in half, and so these reactions now happen twice, one for each molecule of the G3P. So once we oxidize it and rip out that energy, once we extract the energy through substrate level phosphorylation, which we'll get to in a second. So basically, once we make some ATPs, we're left with this final product here of glycolysis. And this here, that's pyruvate. Okay? That's pyruvate. So here's a very overall, here's the summary of it all. And so you can see we start with 1 molecule of glucose, and we end with two molecules of pyruvate. All right, we've added 2 ATPs to the system here, right? And of course, we get left with 2 ADPs. And we took 2 NAD plus molecules, NAD plus, and we got the energy out of the G3P to make NADH. Okay, NADH. Now, it's important to realize here that without NAD+, you are unable to extract the energy from G3P, and so the pathway will stop here. And these things will build up in the cell and be useless, and we will not be able to extract the ATP from it. Okay, that's very important. We'll talk about why that's important when we do fermentation later. Okay, for now, just understand we need this NAD+ to pick up the energy to allow the process to continue. Now, I invoked the phrase substrate level phosphorylation in the previous slide. Okay, up to this point, you've learned only one way to make ATP. If you remember on the thylakoid, right, this is the thylakoid space here, and this is filled with a lot of protons. You'd remember that there's this ATP synthase molecule that can use the protons as they flow downhill to take ADP plus an organic phosphate and make ATP, right? So this is the way we made ATP before. This is what we're familiar with. This next way is a different way of making ATP. All right, what we do is we take an enzyme, and here, I'll tell you what, enzyme with an active site, all right, and we take, so enzyme with the active site, we take a substrate molecule. That substrate molecule, S, happens to have a high energy phosphate group on it. And so this enzyme is going to be able to take ADP and this phosphate and transfer it on over. And so now we have ATP. Okay, so this is a new way of making ATP, and it's called substrate level phosphorylation. And we use this during the glycolytic pathway, during glycolysis, to generate ATP. Now this slide, I have to adjust this slide. This is a little confusing the way it's written. So it says here that NADH mediates redox. Okay, and so what we really mean is the NADH molecule physically shuttles that energy to then deposit it somewhere else to use it. Now, this is why this is confusing. It doesn't shuttle it to the Krebs cycle. No, this is wrong. It shuttles it to the oxidative phosphorylation pathway. Okay, and what that is, that's the electron transport chain. And that's the same idea that we saw in photosynthesis. Okay, and so NADH and FADH2 both together drive these redox reactions that generate ATP. in that last phase. And so when I say the redox reactions, the electron transport chain is basically a series of redox reactions where the electrons are flowing downhill and oxygen's waiting at the bottom to receive it to make water with some protons in there. Okay, so the NADH, the FADH2, their job is to hand their energy off downhill, and so collectively this is the electron transport chain. Okay, this is all conceptual, of course. When you look in the book and you look in the other diagrams and the slides, you'll see how all this actually looks a little more physically. Now for these next three slides, I'm going to actually end up removing these from the final version of this PowerPoint. I think that at this point in the game, understanding the regulation of glycolysis is a little too much. And so what I'm going to do here is just tell you simply that in any metabolic pathway, of which glycolysis is a good example of one, there are ways in which the pathways can go forward and sometimes backwards, right? So think about it. In our bodies, when we metabolize sugar and break it down, we get energy out of it. Our blood sugar drops. But what about times when we have too much sugar in our bloods? Right? We don't want to just keep making ATP. And so there are instances in our lives every day when we want to actually take the sugar out of our blood and put it in the form of, if you remember, glycogen. Right? So we actually want to do glycolysis in reverse sometimes. And so for example, let's say we have a ton of ATP right here. All right? If this reaction here has a ton of ATP built up, it will actually end up starting to push the reaction back that way, right? So that's all I'm going to say about this. You don't need to know the next few slides, and I will not ask you any questions on any tests or quizzes about glycolysis regulation, right? So this slide here, you cannot worry about this, and you cannot worry about this. So in summary, glycolysis starts with a sugar molecule, glucose, and it ends with 2 pyruvates, 2 NADHs, and 2 net ATPs. Okay? And in the presence of oxygen, we're going to then take these 2 pyruvates and we're going to go into the mitochondrial matrix. This next stage, all right, now I call this stage 2 of cell respiration, and the way I like to think of it is there are 4 steps. Wow, did I just do that? There are 4 steps. Glycolysis, the preparatory reaction, the PrEP reaction, otherwise known as pyruvate oxidation, the Krebs cycle, and oxidative phosphorylation with the electron transport chain. The book does not consider pyruvate oxidation or the PrEP reaction as a step, but it really is. It's a separate pathway. So that's why this slide talks about that as stage 2. So pyruvate is transported into the mitochondrial matrix. It is actively pumped. So remember, that's going to need some energy. From here, it is broken down by an enzyme called pyruvate dehydrogenase. There's that dehydrogenase again, right? And so that tells you we're going to take 2 hydrogens and we're going to rip them off of something, in this case pyruvate, and put that energy into an NADH molecule, right? In the process of doing that, we're going to knock off a carbon dioxide and we're going to be left with an acetyl group, okay? So a 3-carbon molecule, there's pyruvate, gets converted to a 2-carbon molecule. One goes away, there's your CO2. And in that bond that we broke, we're going to take that energy and dump it into an NADH molecule. All right? This is what the enzyme actually looks like. They've done some really amazing visualizations on this. This is called pyruvate dehydrogenase. And so basically, this bridges the gap between the anaerobic world and the aerobic world, right? So that's an important point. If this is a cell and this is the mitochondria and this is glycolysis here, and we make pyruvate, this is happening in the cytoplasm, right? this is an environment that does not care if it has oxygen or not. This is the same environment that is present in bacteria and archaeans and everybody in between. The mitochondria is where we're going to be able to utilize the oxygen in aerobic processes. So fundamentally, glycolysis is an anaerobic process, okay? Whereas the Krebs cycle and oxidative phosphorylation and pyruvate oxidation are all technically aerobic processes in the sense of they require the presence of oxygen for them to happen, okay? All right, so the pyruvate oxidation step here, we're going to again start with pyruvate and end with 2 carbon dioxides. Wait, why 2? Well, remember, there are, for each of these pyruvates, there's 2 of them, right? There's 2 pyruvates per glucose. And so starting with a 1 molecule glucose, right, I'm 2 pyruvates. And each one of those pyruvates is going to give off 1 carbon dioxide, all right? And in turn, 1 NADH. So for a total of 2 CO2s and 2 NADHs, okay? So here we can see pyruvate being transported across both membranes. Right, now what's interesting about this is that this transport, these proteins here that bring pyruvate in, are driven by the same osmotic force, the chemiosmotic force that generates the ATP that we're going to learn and see about later. And so this whole inside, the red squiggle area this is the space between the 2 mitochondrial membranes. This is filled with the protons being pumped by the electron transport chain we'll get to later. And so some of that energy goes to making ATP, right, as we'll talk about more, but some of that energy goes to actually pumping pyruvate in. Okay, so we're actually using the energy for 2 different things. From here, so this is called the matrix, remember, from the matrix, the pyruvate is then oxidized by the enzyme pyruvate dehydrogenase, and it is converted to an acetyl-CoA plus, it is converted to an acetyl-CoA plus some carbon dioxide. Okay, so the acetyl-CoA comes in over here, and you can see this SH, that's the sulfhydryl group on an amino acid. That's probably cysteine or methionine. And the key there is that's going to help form a covalent bond between the coenzyme and the remaining 2 carbons that are going to come off of the pyruvate, right? So you can see from here to here, right? All right, so we are taking this portion of the pyruvate, the 2 carbons here, which are here now, right? Those 2 carbons, this portion of pyruvate, when cleaved, will become this. And so this is now just this here. If it was floating around in space, that'd be acetic acid. That's the formula for acetic acid. Okay. Attached, however, to a molecule, it is now a functional group, which we call the acetyl group, right? And so the coenzyme A now has a new shiny acetyl group attached to it. Okay, so that's the gist of this. Okay, the last stage in cell respiration, when it comes to the original carbons of the original glucose, we call the Krebs cycle. Sometimes we call it the citric acid cycle. And what it does is it takes the 2 remaining carbons, right? And remember, there's 2 of them. Right? There's 2 acetyl-CoAs that come in. And so the acetyl group here has 2 carbons, right? And so there are 2 carbon molecules that come into the Krebs cycle attached to the CoA. And what this story is, it's about how we're going to get the last remaining energy out of those 2 carbons. Okay, so it is a cycle like the Calvin cycle in that when we start it, okay, so the very first reaction you can see, don't worry about reading this yet. Here's your acetyl-CoA coming in. This is here already. We're going to attach these 2 things and get that. And by the time we go through all this, we're going to end with what we started. That's why it's called the cycle, right? Oh, as a side note, sometimes the citric acid cycle is also called the TCA cycle for tricarboxylic acid cycle. Okay. All right, so another thing to keep in mind is that this occurs 2 times per 1 glucose molecule. Okay. All right, so just like glycolysis, you're not going to need to know the names of the intermediates or the enzymes. But if you ever are going to go on to take higher level biology classes like biochemistry, you might want to take this as an opportunity to see it once and actually write it all out and understand it, time permitting. Okay, but for our purposes, what I want you to do is know the ins and the outs. All right, so when we start here, here's our acetyl-CoA. All right, we're going to take that 2-carbon acetyl-CoA and we're going to attach those 2 carbons to a substrate that's 4 carbons. Okay, so this is called oxaloacetate. Oxaloacetate. Wow, oxaloacetate. That's what I've always say. Anyway, we got a 4-carbon molecule and a 2-carbon molecule. And an enzyme is going to link these together, and we're going to get citrate, which is why we call it the citric acid cycle. That's a 6-carbon molecule. Okay, citrate, after it gets spun through and becomes isocitrate, then has some of its energy extracted by a dehydrogenase enzyme. There's our first NADH molecule popping out, and as a side product, we have our carbon dioxide coming off. This forms alpha ketoglutarate, which is a 5-carbon molecule with a little less energy. And we're going to do the same thing to alpha ketoglutarate that we did to isocitrate. We're going to take another carbon away, and in that downhill, we're going to capture some energy into an NADH molecule. We're then left with a 4-carbon high energy molecule attached, it so happens, to coenzyme A again. Okay, and so succinyl-CoA is then further oxidized to succinate. All right, and the succinate molecule is then further dehydrogenated to a fumarate molecule. Now here, instead of NAD, this is where FAD comes into play. So we steal some energy out of the succinate, creating the fumarate, and we get FAD reduced to FADH2. Okay, now let's go back a step. From the succinyl-CoA to the succinate, in addition to handing off the 4-carbon molecule from the coenzyme to succinate, or rather, I should say, in this reaction here, from succinyl-CoA to succinate, we are going to take some of that energy using the coenzyme as a shuttle, and we are going to dump that energy into a GDP molecule. This is equivalent to ADP, right? Adenosine dinucleotide phosphate. I'm sorry, did I just say that? This is adenosine diphosphate. They're having 2 phosphate groups attached to each other. This is guanine diphosphate, okay? Or I should say guanosine diphosphate. This, like ADP, is lower in energy, and we're going to rip that energy out of succinyl-CoA, and we're going to make GTP. Okay. Now GTP is not commonly used in metabolic pathways because GTP is way, way, way stronger than ATP. It has a much higher delta G. I believe it's something like 50 kilocalories instead of 7. And so sometimes what the cell does is it takes this GTP and it then converts it to a more usable form, ATP. And then from here we can use ATP for everything. Okay, most textbooks don't even bother talking about the GTP part. So for our purposes, what you just need to worry about is, in addition to the NADs and the FADs here, know the ATP. We're not even going to talk about that, okay, anymore. So let's go back here now. We have succinate getting oxidized to fumarate and making our FAD. Fumarate then, in the presence of water, gets converted to malate. You might remember where we saw this before. If you remember C4 photosynthesis, this is the form of carbon fixation that occurs in C4 and CAM plants. The malate is then, which has a little bit of energy in it, is then finally oxidized to its lower oxidized state called oxaloacetate, which is the starting point, and we can begin the cycle again. In the process of malate being oxidized, we have our very last NADH formed. All right, so let's go back now and look at what we need to know. We start with an acetyl group, okay? It gets attached to a 4-carbon molecule, so the 4 and the 2 come and you get 6, so no citrate, and that's all you have to know here. Then know the products. In the process of going downhill, we have 1, 2, 3 NADHs, 1 FADH2, 1 ATP per cycle. Okay, this is per cycle. Now, because we have 2 of these per glucose molecule, right, from the original, we are going to take the products of this and double it. So the grand total for the citric acid cycle, for 1 molecule of glucose, we produce 6 NADH, 2 FADH2, and 2 ATP. Okay? So at this point, at the end of the citric acid cycle, we now have no more carbon from the original glucose. Okay, so let's do the carbon accounting. There are 2 carbons that are released here for a total of 4, right, for the 2 glucose for 1 glucose molecule, 4 carbon dioxides, and of course, just before the Krebs cycle, we had pyruvate oxidation, which is where the other 2 carbon dioxides are released. So for 1 glucose molecule, we are going to have 6 total CO2s released, right? So there's a conservation of all those 6 atoms of carbon here are still found, and they're just lower energy now. Okay, so that's the Krebs cycle. Here's another version of it. And this is a more bite-sized way of understanding it. You don't have to worry about the names of everything. And really, this is the level of detail that you should know. So here's your citrate and your oxaloacetate, but you don't need to know the names of any of this. Okay. Now you see here, this is outdated now. You see this 30 to 34. Let's say that this is about 30-ish. We can't know for sure. That's what it just turns out it's too variable. And so we say it's about 30 total ATP. We haven't even talked about this yet, so really don't worry too much about this at this point. Right now, understand this slide as understanding the Krebs cycle or the citric acid cycle. All right. So again, 1 FADH2 here, 1, 2, 3 NADHs, 1 ATP, and then the 2 carbon dioxides. All right. And here's another way, the last way of doing it. You can see here in this case, there's another regulation step here. And again, I'm not going to have you worry today about the regulation steps here. So don't worry about this slide. And then this slide here, this just gives you a summary of the entire process. But again, this is more than you know. And so study this on your own if you want, but you do not need to know the names of the enzymes for the purpose of our test. All right, the very last stage, here we are. We are now on the home stretch, and we're going to talk about something called oxidative phosphorylation. The reason it's called oxidative phosphorylation is 1, we're going to need oxygen, and 2, we're going to phosphorylate ADP to make ATP. Okay? So this is the truly aerobic process. This is really the only truly aerobic process that we're going to talk about. And it occurs, as I said earlier, on the little invaginations of the inner membrane of the mitochondria, so on the cristae. And so if you think about it, you can understand now why we have these invaginations. We're maximizing the surface area. The more invaginations we have, the more machines we can fit on the membrane, and the more ATP we can make. Okay? So here we have the basics of it. What this is this membrane right here. So this is a phospholipid bilayer, just like everything we've talked about. We're going to zoom in to say this part right here, and that's what we're looking at here. Okay, so here are your phospholipids. There's your outer leaflet and inner leaflets. You can see now here's a bunch of protons, and so these protons here, these are let's see, where are we here? We're going to be pumping them this way, and so we're pumping them this way. And so this area here, this is the space in between the membranes. Okay, sometimes you call this the mitochondria lumen. Okay, sometimes they call that the intramembrane space. In any case, it is an area, and we have a volume of space in here, and we're going to try to fill that with protons. How? Well, so in the matrix, in this open area here, which is this area right here, we are going to take our high energy electron carriers, our NADH and our FADH2, and we are going to reduce the electron transport chain. Okay, so here you see this area right here. You can think of this box as the ETC. All right, there's all this substructure in there you don't have to worry about. The only thing I really want you to know from this is cytochrome C. Cytochrome C. Cytochrome C is one of the cytochromes residing within the electron transport chain, and in general, and it's a good example of a cytochrome. So cytochromes are these enzymes that help shuttle the electrons and in turn help pump protons. Okay, so you can see this whole thing here as one big proton pump. Protons pumping. So we're going from the matrix into the lumen of the mitochondria, from the matrix into the lumen. And so you can see there's more protons here than here. That's the idea. We're building up a proton gradient here. And then we're going to allow those protons to flow out down this machine, right? This blue thing here, this is ATP synthase. All right. So now that we understand the basics here, let's summarize it by saying it like this. NADH contributes energy to a pump that allows that pump to bring protons into the lumen. Those protons are then, as they build up, used to generate a force in this enzyme, the ATP synthase, which literally spins. And in that spinning process, that mechanical process, it takes adenosine diphosphate and an inorganic phosphate, and it links them together to form ATP. Okay? Now the question is, how many ATP molecules can I make from a given NADH molecule? And so we can kind of do a currency exchange here. So here's how you think of it. For every one NADH molecule, you can pump enough protons into the lumen so that you can make about 2.5 ATP. Okay? For every one FADH molecule, which is a little weaker than NADH, and you can see in this diagram, the FADH is handing its energy a little bit more downstream on the electron transport chain. So instead of starting here, it's starting a little bit down. So it doesn't pump as many protons. The amount of energy here is only enough to make about 1.5 ATPs. Okay, so what do I mean by that? Well, again, the number of protons that I pump in here, there's so many protons that come down here to make an ATP. And the number of protons coming in from one NADH's energy, that amount of proton gives us about 2.5 ATP worth of energy. Okay? And so what we can do there, we're not going to do this right now, but you can add up the X number of NADH molecules and Y number of FADH2 molecules, and you can add them up and do this conversion of the 2.5 and the 1.5, and that's how we come up with about 30 ATP. Okay? And so to summarize it, you could just say that the purpose of oxidative phosphorylation, okay, and by the way, so this is the phosphorylation part. Okay, and where's the oxidative part? Where's oxygen playing a role here? Well, here's oxygen, all right? And basically, what oxygen's doing is this. The NADH and the FADH2 take their electrons, flow them downhill, and when they're cooled off after they've done all their work, here they are, we're going to take some protons, which are floating around here, and combine them with oxygen and make some water. Okay, so oxygen is what's waiting down here to grab those electrons. If oxygen's not here, okay, if there's no oxygen, then there's no way to get rid of these electrons. If there's no way to get rid of those electrons, you can think of those electrons as gunking up the bottom of the transport chain, and that's going to prevent all of this from continuing. Okay, it's all going to come crashing to a halt. And so that's why we need oxygen. All right, so oxidative phosphorylation can be said to couple the oxidation of NADH, right, the loss of energy, the oxidation of NADH and FADH2 to the phosphorylation of ADP to make ATP. So this is just another slide that says the same thing. And you can see, again, there's cytochrome C, there's the buildup of the protons, there's the shuttling of the electrons into the transport chain by both NADH and FADH2. Here's the oxygen, all right, but you don't really need to study this slide any more than the other if you're already getting it. And here's another view of the whole thing. What I really want to get at here, yeah, so this slide, you can see the electrons flowing more downhill to be caught by oxygen. That's what I really wanted to see here, was the oxygens at the end to catch those electrons. So that's the key here. And another concept that this slide touches on is that each one of the members of the electron transport chain is basically able to move the electrons because each member is more electronegative than the last. In other words, this guy here is stronger than this guy here, and so he's going to get the electron. Then this guy is stronger than this guy, and he's going to get it, and they just keep basically stealing it from each other, right? It's like a game of hot potato. That's literally what it's like, because the electrons are like hot potatoes. And at the very end, it's, by the time it gets over here, it's cooled off, and oxygen's the last one to grab it. Once oxygen can pull this out, it allows another pair to come back, or to go in, right? So it's this constant flow that is mediated by oxygen being there, okay? And here's one last view of it all. But again, you don't have to study this slide if you already understand. You don't need to know the names of the different complexes. So I should edit this slide out too. And again, you don't need to know all the details here. You don't need to know all the details here, but there's your cytochrome C. Okay, and by the way, why this one? It doesn't seem to be that important. Cytochrome C happens to be a protein that we've studied a lot, and we have been able to compare its gene sequence across all domains of life. Because everybody has cytochrome C, we can compare the way the gene looks over time. And this is one of the molecular pieces of evidence we have of the commonality of life. All right, now that you understand the basics here, I can now tell you what poisons are. Well, I can tell you what specific kinds of poisons are. So in this case, cyanide. We've all heard about cyanide. We've seen movies where people break open a false tooth and it contains a little cyanide capsule, and they take the cyanide and they die very quickly. What cyanide does is it basically takes the place of oxygen. Right, and so oxygen is going to react with this, the fourth complex, electron transport chain. And so this thing here is the cytochrome oxidase complex 4. You don't need to know that. But basically, cyanide prevents the handoff of the handoff of the electrons to the oxygen. And so it stops the electron transport chain. If the electron transport chain stops, right, if this is no longer pumping protons, then there's no more protons to flow downhill, then there's no more ATP generated. Then you get death. Okay. Another slide. Again, you don't have to see this. I should delete this one. Delete this one. You can just read this. There's nothing added here. These are just redundant slides, really, and I have to just clean this up. And this is, again, just another redundant slide. You don't have to worry about this. This is a good summary. You can read this through on your own. Again, summary. I have to delete these, I guess. Right, more information. You can study this on your own. Oh, I guess one point here is that the protons, because they are positively charged, they are not able to pass through the membrane. They can't do that. They will actually accumulate on the surface here, which is good because then they're ready to go right through this little hole that's made by the ATP synthase. Right, and this process here, I've said it a couple times, but here's the formal word. It's called chemiosmosis. Basically, it's like osmosis, the diffusion of water, but instead of water, it's a chemical. In this case, the proton. Okay. The ATP synthase molecule itself, you don't need to know this. This is just cool. It has all of these subunits. Each one of these subunits is itself a tertiary protein. So this is a quaternary protein. And it works in the same way a watermill does, where it will actually rotate around its axis. And in that process of rotating, it uses the mechanical energy to staple the ADP to the phosphate. It's pretty cool. All right, so if we do a quick summary now of all of the yield of it all, we get to about 30 to 34 ATP molecules. All right, so let's do a quick summary of this. Oh, and by the way, we would say it's rarely achieved. The maximum amount of ATP is rarely achieved because there are other things that NADH does, and there's other things that that proton gradient does, right? You already learned another mechanism of that. That is when you had the pyruvate being transported across the mitochondrial membranes, right? Remember, that required the proton gradient too. So that's why we can never get the maximum. It's just a maximum theoretical yield. You don't need to worry about this slide. You can skip this one. And you can skip this one. Okay. This has to do with the fact that now that we understand the basics of glycolysis, we can now bring in other carbs, the proteins and fats and oils, to breaking down them for energy, right? Remember, cell respiration is just any organic molecule and extracting the energy. And so here's the basic idea. So let's see. So here's glycolysis, here's pyruvate oxidation, here's the citric acid cycle, and here's oxidative phosphorylation. Okay, so here's the stuff that happens in the mitochondria. So when we eat sugars, we already understand that starts over here. So the other sugars, the fructose and the galactose and all those get converted in various pathways to glucose. So you can think of all the other sugars, whatever those things are, funneling down into some pathway to output them all being the same, glucose, which can then go into glycolysis. Okay, when we eat fats, remember that fats, these triglycerides, remember that they have the fatty acid chains, right? The triglycerides. So these fatty acid tails, I remember telling you guys when we first learned this that there's always an even number of carbons on these tails. Now you can understand why. You can break those down and you can generate for each 2 carbons an acetyl-CoA, right? Remember that acetyl-CoA is 2 carbons bound to the coenzyme A. And so a single fatty acid chain can generate 20 or 30 of these things. Okay, so as we break our fats down, we can feed those fatty acids directly into this portion of the respiratory chain, or this portion of the pathway. Okay, and then the glycerol, what's left over, the glycerol can actually be fed into glycolysis and converted probably into pyruvate. Proteins depend on the amino acids. So the first thing that happens is we get rid of that very toxic amino group, the NH2. All right, and so that's why we have the waste product ammonia coming out. This is toxic stuff. This is why our pee is very, very filled with uric acid and ammonias. Urea and ammonia, because we need to get rid of toxic nitrogenous wastes. So we get rid of the amino group, and then depending on what amino acid it is, we either feed it directly into the Krebs cycle, or we will feed it into the later stages of glycolysis. Okay, so all of the foods that we eat, you can now understand, get fed into the process in some particular way. Here's another picture showing the same basic concepts. Everything we just talked about assumes that oxygen is present in order for it all to work. However, the world didn't start with oxygen in it, and there are still plenty of places where oxygen is not in abundance. Think of the very bottom of the ocean, for example. And so for environments that do lack oxygen, or during times when otherwise aerobic situations become anaerobic, so during oxygen deficient times, there are basically two strategies that are utilized for survival. The first is to find some other acceptor of electrons besides oxygen, and the other is to not use oxidative phosphorylation at all, or electron transport chains, but instead just stick to the substrate level phosphorylation. So in other words, just do the glycolytic reactions. So some organisms, particularly those in the deep sea thermal vents, instead of using oxygen in their respiratory chains, they will use other molecules, like in this case we have nitrates, right? So this slide here, this basically shows a similar situation as the inside of our mitochondrial inner membrane. The difference being, so the similarities being, here's your NADH handing energy off, here's the pumping of the protons, here's the ATP synthase making the ATP. The difference is where the electrons end up at the end, the final electron acceptor. In this case, there's no oxygen. Instead, you can see this thing here. That's the nitrate ion, okay? In other organisms, this would be replaced by a sulfate, okay? So those are the ones that happen in the deep sea thermal vents. E. coli, when it is under anaerobic conditions, will use the nitrate. Some organisms will actually use an iron ion, the ferric ion, Fe3+, and still others will use various organic compounds such as DMSO, dimethyl sulfate oxidate, DMSO. I'll just leave it at that. Okay. Now, when we don't use any respiratory chain whatsoever, when there is no phosphorylation occurring with ATP synthase, we have just glycolysis. And at the end of glycolysis, we have our pyruvate and we have our NADH. Okay? What fermentation does is it allows the continuation of glycolysis by taking that NADH, which is now essentially useless, and dumping the energy out of it to make it NAD+ again, so that the process can continue. All right, here, let me show you a picture. All right, so here's glucose, and then here's the various intermediates of it, right, until we finally get down here to, let me say this again. Here's pyruvate. Here's the end of it all. Okay, in this free fall from glucose to pyruvate, we have NAD here to catch some of that energy, right? And then normally, in the presence of oxygen, this would go into the mitochondria. In the absence of oxygen, though, the NADH is not going anywhere. The pyruvate is not going anywhere. And so this is what happens instead. The NADH dumps its energy into pyruvate so that it can now go and get more. So in other words, it can continue this. It can continue taking the glucose and getting the ATPs out of it. Okay, so what happens to poor pyruvate when it gets exploded with all this energy? Well, it depends. It depends on the species. It depends on the genes you have. In humans and other mammals, we go through what's called lactic acid fermentation. And so there are enzymes that take pyruvate and the NADH and convert that pyruvate into something called lactate. Okay? In other organisms, their genes dictate that they will break these down into alcohol, ethanol in particular, and carbon dioxide. And so ethanol fermentation is what we have harnessed to perform things like alcoholic fermentation, to make our beers and wines and ales and all that. Okay, so again, the goal of fermentation is to regenerate the NAD+, so that glycolysis can continue and we can continue to generate this very small paltry sum of 2 net ATPs. Okay, so this is just a summary slide showing you the process, and you can see in this case we have lactic acid fermentation and we have alcohol fermentation. So the advantages and disadvantages of fermentation is what this slide's all about. Now in humans, fermentation under very extreme circumstances can provide us with a vast amount of very quick ATP generation. Okay, so when we go into our fight or flight instinct, or when we have situations where we just need to suddenly give a tremendous amount of muscle action, for example, super fast running, picking up cars to save children, that sort of thing. It's kind of silly to think of that, but it happens. And so ATP can be created very quickly with fermentation. Okay. Also, as we run, as we continue running to the point where we no longer have enough oxygen coming in than the carbon dioxide leaving, we go into what's called oxygen debt. All right, you start panting, you start running really fast, and then you start feeling a burn. Okay, and the old way of thinking of this was that the lactic acid actually changes the pH of the muscle cells and causing a burning feeling. It turns out, at least to my knowledge, lactic acid is not the direct cause of that burn anymore. This is a good example of correlation and causation. Now, I haven't read up on what the actual cause is or if we know about it yet, but I do know that this idea of lactic acid buildup is no longer the direct cause of muscle cramping. Okay, so anyway, that said, what's going on in our cells? When we go through lactic acid fermentation, because we don't have enough oxygen coming in, what happens is that lactate ends up getting pumped to the liver, where it is then converted using energy, converted to pyruvate, which then, in the presence of oxygen, goes and does its thing that we've already talked about. Okay, so we can, for a short amount of time, go into oxygen debt, and then we spend energy to basically equilibrate and get back to our homeostatic conditions. Okay, and here's alcohol fermentation. So there are yeast. Okay, you might not know this, but yeast are fungus. They're fungi. All right, and so they are what are called facultative aerobes. All right, which means, anaerobes. They're facultative anaerobes, which means that under normal circumstances, in the presence of oxygen, they can go through oxidative phosphorylation and do everything that we do. However, in the absence of oxygen, they can go through and produce ATP for very long periods of time under anaerobic conditions. Right? So that concept of facultative means they can do both. They can do aerobic and anaerobic respiration. In contrast to facultative anaerobes, there are strict anaerobes. Oh boy, I can't spell this. Anaerobes. Strict anaerobes are generally bacteria that are not able to be in the presence of oxygen, right? And so these are the ones that evolved first on Earth, and they're still hanging around. If you ever had a puncture wound, if this is your skin, and you accidentally have, say, I don't know, a friend throw a dart into your skin by accident, right? Which I had happen in high school, and it does not feel good at all. This is called a puncture wound. And at the very, very bottom of the puncture wound, this area here, it's very dangerous because this is the site where strict aerobes can infect you. This is where they can grow and infect. Right. The question is, how they get there? Well, it's not that they necessarily die in the presence of oxygen, it's that they just can't grow. And so once you deposit them there, which they might be found at the tip of that needle, because they're found everywhere, they would then flourish and blossom here. Right. This is why things like dog bites are dangerous, or any bite is dangerous. Any wound that goes deep into the skin like that, because this is a breeding ground for anaerobic bacteria. Okay. All right, so this is a little outdated now. Cell respiration, it's not 36 or 38. We just say it's about 30. Okay? And so that's it. That's the end of it. One thing you should do on your own is do some quick accounting. Take the ATP that is made from glycolysis. Okay, there's 2 there, right? 2 net. Take the ATP from the Krebs cycle. Right, there's 2 there. And then, so there are a total of 4 ATP made from substrate level phosphorylation. Okay, and now let's do NADHs. In glycolysis, we have 2 NADHs. In pyruvate oxidation, there are 2 NADHs. In the Krebs cycle, there are 6 NADHs and 2 FADH2s. Okay, so let's do some math now. 2 plus 2 is 4, plus 6 is 10. So we have 10 NADH and 2 FADH2. Okay. Remember the conversion factor we talked about before. 2.5 ATPs can be generated through the protons provided by the transport through the energy of 1 NADH, right? So for every 1 NADH, we can make 2.5 ATPs, and for every FADH2, we can only make 1.5. Okay, so 10 times 2.5 is 25 ATPs, right? And then 2 times 1.5 is 3 ATPs. So that is a total of about 28 ATPs can be generated through oxidative phosphorylation. And then another 4 can be created by substrate level phosphorylation for a total of 32 ATPs, about. Okay? So that's it. You guys we're at about an hour and 12 minutes here, not too shabby. And keep studying, and I'll see you guys next class.